as Rita's just said, I work at over at St Mary's in the MRC Centre for the MRC Centre for Global Infectious Disease Analysis. We're part of the School of Public Health. And I work in teams to try to better understand the transmission dynamics of infectious disease. There are two things about my job that I really like. It's a privilege to work on a topic that, if you get it right, allows you to influence health policy. So if we do a good job and people and policymakers make slightly better decisions, it's great. It's a wonderful motivation and a great part of the job. But the other side of it is the pure science. These are fascinating systems. The interplay between pathogens and their human hosts and all the different processes involved, the actual nuts and bolts of the science, I find absolutely fascinating. So what I'd like to do in the next 45 minutes or so is share a little bit about both of those things with you. So I'll try and motivate the questions, tell you about the answers, but if I can, also explain a little bit about how we do it. And as I said, I work at the MRC Centre, and a few years ago, we were brainstorming how to do outreach. There's a lot of emphasis on scientists now to get out there and try and explain their science to people other than their fellow experts. And what we kind of realized as we were brainstorming outreach is that basically what we do is one massive game of top trumps. So many of you will recognize these cards from when you were young. This is the Predator top trumps. You get a deck of all different things. You have to, everyone who's playing gets a selection of cards. You have to look at the top card. You pick a category, say weight, and then you say, right, I've got the heaviest Predator. And if your opponents have not got as heavy a Predator, then they have to give you your cards. And you carry on playing until someone's just got all the cards. I love these games. They're still around now. I have played Hello Kitty top trumps more times than I would care to admit. And there's a slide at the end that will explain that. But this is a great game. It's a great game because it teaches you about statistics without you even knowing you're learning about statistics. It's continuous and discrete distributions. It's about the expectation, the probability of the next thing that will happen, given what you've seen before. It really is a training in the key intuition for statistics. But it's also great because it generates fantastic conversations. So when you're playing this, you think, well, which animal would I least like to meet? Which is the worst of these animals? What would that animal try and do to me? How would I try and escape? What would my mitigation strategies be with an encounter with that animal? I might not have used that word when I was 11. But so, as I said before, what we do basically boils down to a continuous game of top trumps. And I'm going to shamelessly structure the talk using these top trumps cards, because that was one of the reasons that we made them. And I'm going to talk a little bit about these three pathogens. You can see that we've not got weight and speed and stealthiness. We've got pandemic potential, percentage of cases that die, and then R0. R0 I'll define later on. It's a measure of how transmissible a pathogen is within the human population. And these, I've worked on lots of different diseases. These are all emerging or potentially emerging threats. None of them are endemic at the moment around the world. And smallpox represents kind of the disease of Christmas past. Hopefully that's in our past and is one of our greatest successes. And I'm actually going to spend most of the time on smallpox for reasons that I hope will become apparent. Ebola was something that was underestimated for many years. And it generated a very difficult outbreak between 2014 and 2016 and is now causing a lot of difficult decisions to be made in the Democratic Republic of Congo even today. And there are very few things that you can predict for sure about infectious disease. But one of them is we almost certainly will have to deal with a moderate or severe pandemic of influenza. I'll explain why we can be so certain. And I'll talk a little bit about some of the work that we did trying to improve the way we prepare for pandemic flu. So that's the kind of structure of what's coming and the slightly different questions about each one. So as I said, I want to first spend a little bit of time introducing smallpox and talking about why it's important and why people are still bothered about it. I'm sure you all know that it's been eradicated and maybe we should just not care about it anymore. But it's a great story. So the picture on the left is taken from the 1958 WHO or the UN Health Assembly. And that's Viktor Zhdanov. He's a Russian virologist. This is 4 years before President Kennedy embarked on a process to send a handful of men to the moon. Viktor Zhdanov said in 1958, we can eradicate smallpox. We'd tried to eradicate diseases before and we'd failed. We'd never successfully eradicated anything, but he said that we could do it. And it was a very bold claim. Smallpox was an integral part of human civilization. Humans had spread out around the world with smallpox following closely behind them. It was such a pervasive part of our culture. In some places, you were not even named. You didn't name your children until they had had smallpox and survived it, because names were precious. And the fatality rate from smallpox was so high, it wasn't worth using a name until you knew they'd survived. So this had been an incredible part of our evolution as a population. But there were good reasons to think that we could get rid of it. There was no animal reservoir, so it couldn't go off and hide in the jungle or anywhere else in some other host. We had a good vaccine. There's a whole load of talks about how immunology developed from Edward Jenner and his smallpox vaccine. And crucially, the symptoms were really clear. So the photo on the right-hand side is, during the 60s and 70s, was one of the most printed images on the planet. That's day 5 of rash for a 9-month-old Pakistani baby. And the vast majority of people who got infected had this characteristic rash, so we knew when someone was infected, and that let us do contact tracing with good kind of accuracy. And the other reason we could try and eradicate it from the whole world is some countries had effectively got rid of it. The herd immunity, which has been in the news a lot today about measles, sorry, no measles in the talk today, but the herd immunity against smallpox was very high. With vaccination, it was so high that it couldn't really sustain itself. There would be a few cases in the UK in the 50s, but they would fizzle out. They'd be from introductions. So if we could just replicate that around the rest of the world, we could get rid of it. 22 years later, we were officially successful. This is a copy of the WHO eradication certificate. WHO would really love to write a few more of these. This is the only one that they've written to date. The last wild case or naturally occurring case was in 78, and then it was certified, the world was certified smallpox-free in 1980, 22 years after Viktor Zhdanov had suggested we get rid of it. Economists have rated smallpox eradication as the most successful global public investment ever. You can add up the money that we don't spend on vaccines and the cost of the lives of the people who don't die from it anymore, and if you put that into any kind of investment framework, the return measures you get are astronomical. So this is a fantastic success, but it's dependent on it not coming back. So why do we still worry about smallpox a little bit? And there are a number of reasons for this. Firstly, vaccination has stopped. I am one of the older people in the UK who wasn't vaccinated. I don't have the bump on my shoulder. Some people will still have that, but when smallpox was eradicated, vaccination stopped in the vast majority of places pretty quickly. There are officially 2 sources of the virus still left, one at the CDC in Atlanta, the US CDC, and one at the equivalent in Siberia, the Russian equivalent of the CDC. The CIA in 2002 said that there were probably at least 3 other sources out there that had been stored without appropriate authorization. They had good evidence that North Korea, Iraq, and France were maintaining stocks of the smallpox virus, and there are probably other ones out there. But that's not the reason it's become a bit more of an issue recently. You might have had some talks or seen a lot about reverse genetics. And the genomes, the genetic sequence for these viruses are very small. They're around 10,000 base pairs. And it's now relatively straightforward to reconstruct that size of genome from just knowing the sequence. And the reason that smallpox is in the news again recently is a group has made horsepox from scratch using entirely standard reverse genetics techniques. And they've tested and shown that it's got the same properties as horsepox in the wild. And that's led everybody to start thinking again about what might happen were there some kind of accidental or perhaps more sinister release. So that's one of the reasons. That's why we think a little bit about smallpox at the moment. what I want to do to try and kind of pull all the bits together is I want to talk about specific questions, and that's kind of how we work. So I mentioned that it's great that we get to understand this cool science, and we kind of look at it through the lens of potential interventions or potential strategies. And when it boils down to it, we get given very specific questions that we have to try and address, and that's what pushes the science forward. So I'm going to kind of use just 3 questions to try and pull it all together. So for smallpox, and this is partly because I happen to have written a paper on exactly this scenario. That's the other reason that we're... So the question is, if there were 10 cases wandering around central London today of people infectious with smallpox, how worried should we be? If we had a stockpile of vaccine, should we run out and vaccinate the whole of the UK or as many people who are close to cases? Would be a very relevant question, and that's kind of come up in the literature, and it's something that we've worked on. So that's our illustrative question. I'm going to leave the smallpox story there and then come back to it at the end. So just before I kind of move on with the next case study, I want to make a few general comments about kind of what we do and how we do it. As I said, there's all different ways you can geek out on this stuff, and I don't want to bypass the actual nuts and bolts of how we do it, because that's one of the things that kind of gets that's really attractive about it for me. So chronic disease, and there's lots of good reasons to study chronic disease, but it's fundamentally different. Your risk of developing a chronic disease is a function of lots of small decisions that you make over the entire course of your life. It's down to your genes for a lot of conditions, it's your diet, it's your exercise lifestyle, and all kinds of things, and it builds up over time, and then you may or may not suffer from the disease at some point during your life. Risk changes slowly, and we intervene in a way that reflects how your risk changes. It's about educating young people, it's about getting your kids not to have too much red meat and things like that, which is difficult when you're barbecuing. But the infectious disease risk changes in a very different way because we catch it from people who are infectious. And lots of infectious diseases operate on short timescales. So your risk of infection from an infectious disease can change very quickly over a short period of time. And that's what makes the maths and the computation fun. So, or interesting and different from chronic disease. So this chart, and this is the first, there's more pictures and words than there are charts, but there's a few charts, and a lot of them do look like this. This is an incidence chart. So we're saying this is for influenza-like illness in the UK, actually, in England and Wales, and it's showing 3 different flu seasons. The week numbering at the bottom is a bit strange because it's a winter epidemic, so it starts in the late autumn and kind of goes through the end of the year and then through into the next year. And the infectious disease is so kind of intriguing because your risk is proportional to the number of people who are infectious. And that changes rapidly over a very short period of time. So in 2010-11, if you look at week 46 and compare it to week 52, you've got about a 20-fold change in your risk of getting infected with flu because there are 20 times as many. And it's that feedback cycle. As more and more people get infected and become infectious, the epidemic grows and grows until something causes it to peak and then it decays. And there's lots of maths and computers around that simple process. But basically, we have to there's no point using standard techniques to represent this. We have to try and represent those mechanisms with maths or with computers. Okay, and then this is kind of how we actually do it. This chart on the left here is an RStudio window. I'm looking around, I might be the only person. I have seen someone on the train into London actually use this. It's not as rare as you think. This or similar things in Python is how an awful lot of analytics gets done these days. And what we do is we spend an awful lot of our time either designing this kind of code, managing it, thinking about it, producing results from it and interpreting it, or hopefully if you've got a few people working with you, you're spending a lot of time talking about it and interpreting it. And then a little bit of your time kind of going out into the field and gathering data and kind of meeting up with policymakers and trying to communicate your results. So I can't really go through the nuts and bolts and explain how the individual code works. I'm not going to try to, but you can think about it really just like a computer game. So it's the problems we work on are very serious. I'm not kind of going to trivialize. There are stressful and important decisions that sometimes influenced by the work that we do. But in terms of the actual creative bit of making a model and using it to generate evidence to inform policy, it's very much like designing a computer game. And they're really, really simple computer games. In fact, for Outreach now, we've been working on a board game that uses a very kind of similar setup and dice to try and explain to children how these models work. So just like a computer game, you'll represent your infectious people in the model maybe with a red dot, and then the susceptible people are the green dots, and then you put them into your computer or your maths, and then you use what you know about the pathogen to say how people get infected. On average, how many new infections will there be? How far away will they be? What type of people will they be in? It's fairly simple assumptions that are relatively easy to put into the computer or to put into maths. And then once you define those assumptions, you let it run forwards, and it will show you who gets infected next and next and next. And then at the end, it will give you an answer. So it'll say how many were infected in a certain time, how many you expected to be infected by the end. And at that stage, we'll try lots of different assumptions and different models, and we'll compare it with data. We won't just pick a first set of assumptions and then say that's how it's going to be. Or we certainly don't do that anymore. We use all of the available information, compare it with the observations, try lots of different variants, and then using the best model or the best set of models, we try and say, right, what might happen if? And a lot of the what if questions that we do are around interventions. So we make some simple assumptions about how each generation progresses from one to the next, but then we can say, okay, if you vaccinate half those people, how will it progress? Or say it was measles and suddenly you haven't got as many immune people as you hoped you had for measles because they've not been vaccinated, then what might happen next? So we combine kind of, if you like, operational kind of logistics and control theory of knowledge of the interventions with these models to try and to be able to run what-if scenarios. And although the coding, and sometimes you solve it with maths and you don't even need to use a computer, you can solve it with a pen and paper sometimes. Other times you need a computing cluster to run for days and days and days. But the solving it part isn't kind of really the difficult part. The difficult part is figuring out what the important bits of the process are and piecing them together. Okay, so with those general kind of comments in mind, I want to do a couple of small examples before I go back to smallpox. And one of the reasons to do this, it's not just because we have the top trumps cards, it's because we can build up the kind of complexity of the model we're using and show a little bit how it translates into actual policy. So, Ebola was underestimated kind of for a long period of time. We discovered it, I think, in 1976 in the DRC, what's now the Democratic Republic of Congo and Sudan, and it caused lots and lots of small clusters. And then in 2014, it broke out in West Africa and was kind of rumbling on for a long time without people paying a lot of attention to it. And then suddenly the cases started to increase very rapidly, and the outbreak was very suddenly much bigger than any outbreak that we'd seen before. And there was an awful lot of debate about how much we needed to respond. So governments around the world were scrambling to decide how much money do we put aside, how important is this, how bad could this be? So that was one of the questions that we worked on. As I said, it was discovered in '76 in the DRC. The main host is almost certainly the fruits bat. You do get Ebola in non-human primates like gorillas, and from gorillas it can cross over to humans. So humans that hunt gorillas and other primates, and that's called bushmeat, will sometimes get infected. And up until 2014, the secondary transmission of human to human was quite contained and rare. And you can't get Ebola from kind of coughing and sneezing in the same way you can get flu or smallpox. It's direct contact with bodily fluids. So there's a lot of transmission while you're caring for patients with Ebola. And then in West Africa, there was a lot of transmission in the preparation of bodies for burial. And when we were working on this, this is how the data looks. And it's a similar format to the data I showed earlier for flu. The number of new cases per week is on the vertical axis, and then it's an epi week again. The week of the year is shown on the x-axis, and the end of these data is around September 2014. And it's increasing alarmingly quickly. And as I said, it's much bigger than any previous outbreaks. So there was a lot of debate about how bad it might be and how serious we needed to take it. How much should we invest? So this is a nice example in some ways because there's a really straightforward set of assumptions that we use. We don't even need to think about susceptible people. You just imagine a very simple model where you've got one infectious person and they undergo a renewal process. This occurs in other parts of science as well. You can see it in forest fires and nuclear explosions. And a renewal process is when that first infectious person generates, say, 2 new infections, and then they go blue. They stop being infectious. And then each of those 2 generates another 2 infectious people. So you've got 4 in that generation, and then 3 people who've recovered. And then those 4 see a very similar, they undergo exactly the same dynamics and produce 8 in the next generation. And that process has has one really strong characteristic, is that it has a constant doubling time. So regardless of the average number in each generation, if each generation is kind of renewing itself in the same way, and on average there's more than 1 new infection in each generation, then the doubling time will be constant. And one really straightforward way you can look at these data, it's the same data or very similar data, and this time they're plotted on a log axis. So on the vertical axis here, rather than going up in kind of 10, 20, 30, 40, it goes up in from 1 to 10 to 100 to 1,000. And if incidence data looks to be on a straight line on a vertical axis, that's really good evidence that a renewal process is underway. And you can see across the 3 different affected countries there, for a considerable period of time, looking at the past 6 weeks at the point that we did it, that was pretty good evidence that it was a consistent renewal process. And that meant it was reasonable to extend the line on the right-hand side to make a what if nothing changes. We had no good reason to think anything would change. And then you plot the whole thing again on a linear axis. So this time we've gone back to going up in 0, 1,000, 2,000, 3,000. And that's the other term that you hear associated with this type of process is exponential growth. And you might recognize that kind of takeoff trajectory as an exponential growth. So we had good evidence, and we could say if nothing changes, this is what we think the time series of cases will be. And so that was basically used. So the answer to the question was, we should invest as much as we possibly can in stopping this outbreak, because even 3 or 4 weeks of growth at that kind of rate would significantly change that part of the world. And had it reproduced that pattern elsewhere in Africa or beyond, it would have absolutely decimated. I mean, it did decimate the 3 economies involved anyway, but it would have been a huge, huge deal. So the answer was, we should invest as much as we can. Very similar charts to these were actually held up during the debate in Congress in the US. The Obama administration asked for $6.2 billion to be allocated. Congress approved something like $5.3 billion. It wasn't all spent. It was just allocated in this emergency process that they have, and governments around the world mobilized as much as they could. Now, the numbers were pretty bad, but they were nowhere near as bad as they could have been had that process continued. So it's difficult to know how much that massive increase in awareness and the actual help that they provided was in changing that process, or whether it might have changed anyway for reasons that we don't fully understand. But there is no doubting the massive increase in awareness and resources that came at exactly the same time that it stopped being growing exponentially in the way that it did. So, although that's controversial, a little bit controversial, it's still relevant today. So it's very relevant today. I am not working on this at the moment. I have rotated off Ebola. But we do have members in the School of Public Health that are actively involved with helping WHO on this outbreak, but these are not proprietary data. This is taken from the web, and it's the latest situation report for Ebola in the DRC as of 14th of April. And you can see that it has been this goes back to April last year, and these are weekly incidents now. You can pretty much ignore the colors, it's just the red are the confirmed cases. And you can see that it picked up and it's maintained a very constant signal with the odd spike, but since February this year, it's suddenly started to show that dramatic uptick that we observed in West Africa. The other thing that I'm not going to talk about much today, and we spend a lot of our time just going over the numbers and cleaning it and understanding the reporting. But the last few weeks when it's confirmed cases are difficult to interpret because there's what you call backfill. So in 3 or 4 weeks' time, when all of the reports for these dates are in, we're not really sure exactly what these two bars will look like. So right now, we're in almost exactly the same situation we were, just at a lower level to the September 2014. And people are looking at this and trying to see direct evidence on the ground for a kind of exponential takeoff. So, okay, right. So moving through to the last example before we return to smallpox. And as I said, pandemic influenza I've done a lot on this over the years, and I now spend a lot of my time actually working on seasonal influenza, motivated mainly from studies that I've done on pandemic influenza. And it has this very high potential, the highest that we gave it in our top trumps distribution, that we almost certainly will see a pandemic in the future. And then the really interesting thing, it's all interesting, I guess, but on the top trumps card, the noteworthy statistic is the percentage of cases that die. And you can see the number here is 0.01%. And that's actually our best estimate for the number of people who got infected in 2009 who died as a direct result of their infection. But the number for the 1918 pandemic is very different from that. The number for the 1918 pandemic is probably closer to 2%. So it's a 200-fold difference between the fatality rate in 2009 and the fatality rate in 1918. So the reason there's so much interest in flu, and the reason there is an entire field of studying flu, is because 100 years ago today, everybody was hearing about flu for the first time because it had just undergone this terrible, terrible pandemic. And it's very likely at some point, we don't know when, but at some point in the future, we will likely have a similar episode. And this is the reason. I think, what we do, infectious disease dynamics is like a bit of a mishmash of lots of different biological fields, and then we kind of glue it together with the maths and computers. So there's a bit of virology here. And you might have heard of H3N2 or H1N1 as the names of flu strains. So H5N1 is a dangerous strain in China and Southeast Asia, and H7N9. And those H and N numbers, they correspond to the outside of the virus. The blue on this schematic are the hemagglutinin, the H's, and the green are the neuraminidase. And because they sit on the outside, that's the bit of the virus that our immune system can recognize. And because of the pandemic, we've been tracking flu for 100 years. Even before we could do genetic sequencing, even before we decoded the genome, we had other techniques to track strains of flu. And we've known about all the different strains that have gone through the human population from looking at people's blood. And the thing we know for sure is that every strain of flu that we've ever seen, we can see in wild birds. So all the evolutionary action for flu happens in the wild birds. Everybody else is really just kind of spillover or dead ends. We're really the dead end of the dead end. The evolution happens in the wild birds. It goes into domestic poultry, into swine, and other kind of proximal hosts that we interact with. And then very occasionally it will spill over into humans. So unless we manage to find a way of wiping out flu in wild birds, and the only way we could do that is probably wiping out wild birds, we will always be challenged with these new strains. And that's why it's the bogeyman of infectious disease with very good reason. And as I said, the 1918 was probably 200 times more severe than the 2009 pandemic, and it generates these kind of pictures. This is a classic pandemic picture, a converted gym in the US with service personnel being isolated because they're infectious, trying to give them some kind of therapy and taking them away from people who they might otherwise give it to. So this is active, this is an isolation intervention. And it's also really evident in the data. This is a chart taken from a paper at the turn of the century that shows all-cause mortality, every way you can die, all the stupid ways to die in the kind of horrible histories type way. So it's all-cause mortality is the top line. It's the rate per 100,000 per year. The blue line is due to non-infectious diseases, and the red line is due to infectious diseases. And that's at the resolution of a century. You can see a great big massive spike in 1918. And that's the reason that there's so much interest in flu. And that's why we constantly plan for what we might do during the next flu pandemic. And what I'd like to do is talk about one specific intervention that we worked on. And there was a lot of interest in preparing for the next pandemic in the early 2000s, partly because of the 9/11 attacks. So everybody was surprised and taken aback at this very unforeseen event. So there's a whole load of horizon scanning. Governments and think tanks around the world did all this horizon scanning to say, what else have we missed? Why didn't we see this coming? And very soon they said, well, terrorists might use biological weapons. And then they said, well, that's not very likely. Mother Nature is probably the greatest threat that we've got, and we need to improve our plans. So they started making, in the early 2000s, lots of different preparedness plans for the pandemic. And one of the big questions was how much they should use quarantine and isolation. So quarantine is when somebody might be infected and you ask them to go off to one side or to stay away from other people in case they become infectious. And then isolation is when you know someone's infectious, that you isolate them and stop them infecting other people. And to do that on a population scale would be a massive deal. You might have to you'd have to ask people to stay home if they've been exposed, not go to work, not generate all the good things that they do at work. And when they were isolated, you probably have to provide somewhere for them to go to be isolated away from their family so that they could protect their family. So it'd be a huge deal if we tried to do it. And what people were interested in is how effective would that be? Would that really make a difference? Because we can't make a vaccine quickly enough in a pandemic. Antivirals aren't that great, and we can't make enough of them for the whole world. There aren't that many things that we can do during a moderate and severe pandemic that we think would have a big impact on the progression of the epidemic. So we and lots of other groups looked at this in a very similar kind of modeling framework. And this time, you've got to kind of locate the susceptible people in their own individual households in these rectangular boxes. And then it's the same kind of idea. The key thing then is that you're much more likely to give it to people in your household. And you have very weak connections to people in other households. But there's lots of other people in those households. So it's still going to get quite a few infections going that way. And you feed those assumptions into the model, and you can run it forward in exactly the same way and predict what the patterns would be in households during a flu pandemic. And then crucially, those interventions like quarantine and isolation, you can express them as changes to those links. So if you quarantine people effectively, then you reduce their ability to infect outside of the household. And if you isolate them when they're definitely ill, then you can massively reduce that within household transmission. Those assumptions are pretty robust if we could stick to it. And even if only 50% of people did adhere to this type of intervention, then the results were very positive. So the black line is a good estimate for the number of infections that you'd get actually on a daily basis as a percentage of a population during a flu pandemic. And the area under that is about 70%. And you can see that with the gradual increasing of quarantine, isolation, possibly using antivirals, you can massively reduce the area under the curve and push it to the right-hand side. And you're talking about reducing the number of infections from, say, in this scenario, 70% down to 30 or even 20% of the population at only 50% adherence to these policies. So this became a mainstay, a little bit of pandemic planning, and it's still in all of our plans. The UK ranks pandemic flu as the #1 non-military threat to public, to the British public. So the answer is yes. It would be difficult to do, but it's certainly been written into all the plans. Okay, so coming back now, built up kind of different bits of the modeling, coming back to kind of the original question and thinking about that scenario where there's 10 cases of smallpox in central London. Should we try to vaccinate everybody? Should we try to vaccinate people within so many kilometers of a case? Let me just check, 1 second. And it's worth thinking about, for this scenario, it's worth thinking a bit about what the default would be. So almost all of our language for infectious diseases actually comes from smallpox. So contact tracing, isolation, quarantine, a lot of those words were originally developed for smallpox over the years. So we would definitely contact trace. And if we had vaccine, we would give vaccine to anyone who was at high risk, who we know had been in contact with the case. Okay, so we would definitely do that and would use the vaccine for that. But the big question is, would you go out and kind of, in a non-targeted way, offer the vaccine to hundreds of thousands of people living around where there were smallpox cases? Some people thought you should, some people thought you shouldn't. And it was a very kind of active debate. So basically, we can take that same concept that we had for the flu, where we've got people in households, we have household transmission, we have between household transmission, but we're not imagining the whole epidemic going through the UK in a kind of in an unsynchronized way. We're thinking about 10 people in central London having this, and then it's spreading out from central London. So you have to pay much more attention to how you link the households together. And the way we did that was by looking at how people travel, their reported movement, commuting, journeys to school, and then making a model of 60 million people in the mainland UK and linking those households in the way that we think is consistent with all those data that we have on how people move, and then setting off the 10 people in central London at day 0 and really hoping that the YouTube connection works. And then we could play out the model of what we thought might happen. And we don't really know how transmissible smallpox would be, whether like the best case is on the left-hand side with an R0 just around 1.5, not that transmissible, then medium transmissibility in the middle and high transmissibility on the right. And eventually, day 39, day 40, it does start to spread out. In the worst case scenario, it's getting out into kind of the Midlands and spreading down into Kent. But it's still just a handful of cases, and this is 43 days after those people first started wandering around infectious. That's a long time, and this is with absolutely no interventions. So the first point to kind of observe about this is it would be really slow. It would be a huge news story, admittedly, and it's not like we would do nothing about it, but it would move really slowly, even in the worst case scenario. And then this is like we sometimes use the phrase in silico science. We've got this system now, it's up and running, it's the best we can do. It's not perfect, it's not a crystal ball, but we can now run experiments on this system and use those to say what we think might happen. And so what we did is we looked at and here I've presented results from kind of the medium, the best guess scenario, not the best case and not the worst case. These are results for kind of the best guess scenario for smallpox. And if we just successfully isolated people, so not even any contact tracing, no vaccination or anything else, if people, when they recognized they were infectious, just went off and were by themselves, that would still contain the outbreak in 89% of simulations. and you'd only get 91 infections, and the whole thing would last for 212 days as our best guess. As I said, this is not a crystal ball, but this is using all the available evidence and putting it together in a model. That's our best guess. If you add into that contact tracing, vaccination, quarantine, and isolation, everything with reasonable parameters that we know from kind of other public health processes, you control 100% of the simulations. Not one does it become a large outbreak. Your average number of infections is down to 22. 10 of those are the original seed cases themselves. It lasts for 84 days, and you only need to use 410 vaccines. Okay, so again, 84 days of knowing that smallpox was circulating in the south of England would still be a big deal, but the actual health impacts would be very, very low. And then, but what people would clamor for, from not just my own kind of uninformed intuition, but from evidence of pre-eradication outbreaks in New York and Yugoslavia, what people would clamor for would be vaccinate everybody who lives near here. So because we've represented this in space, and we can go and have vaccination queues, and it's just like a game, not a frivolous game, but it's just like a computer game. We can run spatial vaccination, where if you're within 15 kilometers of a case, we put you in the queue for vaccine, and we imagine how many people at our best rate we could vaccinate. And at the 15-kilometer resolution, you'd use 9 million vaccines. And we estimate that you would avert an extra 4 cases with those 9 million vaccines, and you'd save yourself 14 days. There are probably economists that would look at the 14 days and say, yep, that's worth it. But 9 million vaccines, given that the crude fatality rate is 20 to 30%, and the crude fatalities associated with vaccination are 1 in a million, it's probably higher than that. You would probably end up killing more people with that vaccination program than you would save by the averted infections. That's the best guess. So the answer is no, you wouldn't try and vaccinate everybody if there were 10 people running around central London for whatever reason, infectious with smallpox. So it's a happy ending. But, and this is where the but comes in, this isn't the only study. There's lots of other evidence that's in pretty good agreement with this. But what would be really interesting is the political dynamics when it happened. And that's something that we can't control. So you get lots of interesting discussion of this when you communicate it to people who would issue the advice or who would work with the politicians to make the decisions. And it does show how the scientific evidence is only ever one part of the puzzle. Okay, so that is pretty much the end of the examples. I've got kind of a few closing comments. Firstly, kind of an apologies for there's a doom and gloom element to this. There's no school parties in today, but there are some children, not all of them related to me, but there are some children. And it's, I'm kind of resentful for growing up in the 70s and everyone talking about air raid shelters and imagining where the nuclear bomb would fall and where we would run to and stuff like that, because it was really unjustified and really anxiety-provoking. I think even the worst scenarios among these, you should have some investment, you should have some people thinking about them, but they're not really major concerns for your everyday life. There's really interesting science, and in the event that these things happened, it would be important. But this underlying science is really interesting, and we shouldn't kind of emphasize the doom and gloom just to get funding too much. Although it's a bit of a niche science, so that we have our own journal. It's called Epidemics, the Journal of Infectious Disease Dynamics, and the publisher Elsevier refer to it as one of their nicest niche journals. So we're a growing bunch of people, and we're well formed, and we have our meetings. But there's a few hundred, maybe 1,000 of us. But it's kind of it's an example of a much broader kind of type of endeavor. So studying the natural world, using mechanistic models, making as many model variants as you can, using the best observations, the best data that you can. That's a real growth industry, and it's very similar to the analytics that are done now in lots of tech companies and in lots of healthcare. So The specifics of what I have talked about are really quite niche, but as we progress through time, the general approach of how we work and the type of questions that we ask is a much bigger kind of endeavor. I guess the final thing is the game of top trump. So which one would I worry about most? What would I do? What do I really think? I spend most of my time working on seasonal flu now, and that, as I mentioned before, is mainly because there's no getting away from it. There will be a pandemic of influenza. There's been one already in our lifetime. There's a reasonable chance that it could be severe. So not to go back to the doom and gloom, but if I have to play top trumps with the diseases, even though it's kind of the bogeyman that everyone knows, pandemic flu is the one that concerns me most. So that's pretty much all I have to say. I have one thanks to give, because I very rarely get to present with my family in the audience, but they are here today, so I only get to do this fun stuff and have a nice, loving family, because they support me so well. So I will thank my family as my last acknowledgement. And thank you very much for your attention.